All right, hello everyone. How is everyone doing today? I hope everyone's doing good. We're so excited to have you with us today. Just before we get started, I just want to do a quick check with everyone. If everyone can see and hear me clearly, do you mind just saying yes or a thumbs up in the chat? Okay, perfect. I see some people saying yes. All right, okay. So, just to get started, welcome to our event, The Science Behind Autism with Ben Ryan. My name is Anilpa. Oh, perfect. I see more people saying yes. This is good. All right. Okay, so welcome to our event. My name is Anilpa. I'm part of the SciMed in Action team, and I'm going to be your host for today. So we're so excited to see that some of you guys are coming in from different countries and time zones from all over the world, including, Germany, Indonesia, India, and the US. So whether you're watching this at midnight or maybe early in the morning, we're so glad to have you with us. So if we can just go to the next slide. What we wanted to do right now, for those of you who may not know, is a Canadian tradition, which is called a land acknowledgement. So we acknowledge that the land we are hosting on is on the traditional territory of many nations, including the Mississaugas of the Credit, the Anishinaabe, the Chippewa, the Haudenosaunee, and the Wendat peoples, and is now home to many diverse First Nations, Inuit, and Métis people. We also acknowledge that Toronto is covered by Treaty 13 with the Mississaugas of the Credit. So now we wanted you guys to get to know a little bit about our organization and about Ben. So SciMed in Action is a pretty recently established organization. We were established in April, and recently we just all came together as a team as of September. And our main focus throughout this whole process was all based on opportunity. I think we all know 2020 has been a tough year for us all, so I think, what we found is right now we have less access to resources and opportunities in our interests. And that's why we at SciMed believe that learning should not just be limited to the classroom. We want you guys who are interested in science and medicine to be able to find opportunities to further your knowledge and help you guys apply that knowledge to the real world. Some of our past initiatives include working with the organization STEM Empowerment and running an event with the scientist Dr. Barrett. However, today's event, The Science Behind Autism, is our first event as a team, which is super exciting. Our goal with you guys today is really just to build a community where we can give you insights, knowledge, and opportunities in the fields of science and medicine that, again, go beyond the school curriculum. So one really great way of gaining this knowledge is by consulting experts. And so today we're very lucky to have with us the neuroscientist Ben Ryan. Ben is a Neuroscience Ph.D. candidate. If we can go to the next slide, perfect. Ben is a Neuroscience Ph.D. candidate and Presidential Fellow at the SUNY Buffalo, where he works with the distinguished professor Dr. Zhen Yan. In his research, Ben is currently investigating some of the molecular mechanisms behind aspects of autism, which he's going to be speaking to you guys about today. He's also the founder and president of the Aspiring Scientists Coalition, which is a community organization that's free to join that provides guidance to students in science. All right, so you guys are in for a really cool presentation today. Ben is going to be taking you through an introduction to developmental disorders, the pathophysiology of autism, and some of his research on mouse models. So during today's webinar, if you have any questions at all for Ben, please feel free to comment them down in the Q&A section. If you guys can see your screens at the bottom right, there should be a bar that says ask a question. So again, if you have any questions for Ben at all, please comment them down below and we will get to them towards the end of the presentation. All right, so without any further ado, let's get it started.
Thank you so much. First off, can you hear me?
If everyone can comment in the chat, I think I can hear you, so I think we're good.
Okay. Few things. First off, I want to say I've never seen the land acknowledgement done before, and I thought that was awesome. I thought it was beautiful, and I really like that. Second, 2 disclaimers. First disclaimer, and these are for the audience, is that one, since I'm presenting the PowerPoint, I can't see my video or your video, so if there's any questions or anything, and Alpa's going to read them to me and let me know. So she will have full privilege to interrupt me and let me know what's going on.
Yeah, I'll be taking care of that.
Awesome. The third thing is that I want to have a disclaimer. I am not a doctor, so I do not work with patients with autism. I don't actually see patients. So I do research regarding autism and the neurobiology of it in mouse models. And a lot of the information that I'm going to be presenting today is based off of what I've gathered from reading papers and textbook knowledge and speaking with other scientists at conferences. So just wanted to get that in the clear. But if you do have questions, of course, please ask. So autism spectrum disorder is a neurodevelopmental disorder. You can assume from that. So neuro is brain, or brain and spinal cord, the nervous system. Developmental, it's a disorder of the brain's development. So it's not the only developmental disorder. We also have intellectual disability, ADHD, and schizophrenia. And if we look at the prevalence of these, you might be actually a little bit surprised that autism affects 1 in every 54 children. That was just raised from 1 in every 59 this year. And intellectual disability is very similar. ADHD is more prevalent, almost more than twice as prevalent, and then schizophrenia is much less prevalent. So what exactly is autism? There are 2 diagnostic criteria of autism. The first is what most people are familiar with, and this is persistent deficits in social communication. So those who are on the autism spectrum often they find it challenging to engage in social interactions. Another less well-known phenotype, shouldn't use scientific words. Another less known diagnostic criteria for autism is the restricted and repetitive patterns of behavior. So kind of a good example of this that I learned from a pediatric psychiatrist that I was working with is that a child with autism may have a bunch of toys and like to organize them, and from smallest to largest, and kind of set them up and then repeatedly organize them. So that's just an example that I like to think of that kind of helps you understand exactly what this looks like. So the average age of diagnosis is 4 years old, although their diagnoses do occur much earlier. There's screenings done at 12, 18, 24 months. You can diagnose a very young child with autism. And typically, sometimes this has happened where the mother starts to notice that her child is not reciprocating eye contact, or gets anxious during social interactions. And the way they essentially screen for autism these days is with something called ADOS, which is the Autism Diagnostic Observation Schedule. So notice the word observation. I've seen this done. I went to a autism diagnostic center, and it's actually an observational assessment where a child is put through a series of tasks. And for example, one of them that they do is they'll give one of these cards at a time and say, can you tell me what emotion is this person expressing? So I'm not a doctor. I'm not going to talk too much about this, but it is interesting the way it's diagnosed, because it's a spectrum. That's a very important thing to keep in mind, is that what the doctors told me is once you meet one person with autism, you've met one person with autism. It's not like everybody who's on the spectrum has very similar behaviors. It's very diverse. So, which is why observational assessment is required, rather than just, you can't just tell, looking at someone or speaking with them for a minute. It's very complex. So a few other things about autism. There's 4 males that have autism to every 1 female. And this is totally not understood. There's really no explanation for this. And this is actually something that I'm a bit curious about investigating in my personal work. But it's a great question of, why are there 4 to 1? So that's, an ongoing question. If you want to get into research on autism, that's a huge open question. Now, another interesting thing about autism is that it has very high comorbidity. Now, and by the way, I'm assuming that the audience I can't even see you guys, but I'm assuming that the audience is coming in from maybe high school age. And throughout this presentation, I'm going to use a few words that you might be like, I've never heard of that before. And the purpose is I want to define them so that you can potentially add these to your vocabulary, because it's cool to have little science terms. Then comorbidity is overlapping. So if I have asthma, and let's say I also have schizophrenia, those are now comorbid disorders. And intellectual disability is seen in almost a third of children with their patients with autism. Epilepsy is 1 in every 4. Over half of those on the spectrum have sleep issues. Another big question of why that happens. Gastrointestinal symptoms, and then also motor difficulties. So in the bottom right, we have this puzzle pieces fitting together, and it's just kind of interesting. It just shows, how interactive autism can be with other similar conditions that, again, not very well understood. So let's talk about the causes of autism. Now, one of the most common causes, I think genetic risk factors account for about 60% of all cases of autism, give or take. It's probably around there somewhere. So the majority. And there's something called de novo gene mutations. Now, again, I'm going to define this. So de novo is Latin, basically just means new. So this means randomly occurring, essentially. So if I have a child and I don't have a mutation in a specific gene, but my child is born with that mutation, it occurred randomly, that's a de novo mutation. If I had the same mutation and I passed it along, it would be inherited mutation. So there are hundreds of genes that are known to be associated with autism. And I just want to also clarify that a lot of people think of like, oh, you have the autism gene. It's not that there's a gene that is associated with autism or a gene that's associated with depression or things like that. There are specific genes that when they are mutated through like substitutions, a gene's mutated, whatever, it's losing its function. And now it's not performing whatever function it's supposed to in cells. And this somehow affects some neurobiological process to result in whatever disorder. So here we can see a lot. There's metabolic disorders, schizophrenia. Many of these things are genetic. And that's essentially how it works, that you lose the function of this gene. And this is also showing that there's a lot of overlap. So whatever, this is just a graphic I pulled from a paper. Now, rather than single gene mutations, there are also something called copy number variations. So you're probably looking at this list of numbers and letters and thinking, what is this? 16p11.2. This is what I'm going to be talking to you mostly about with my personal work, is chromosome 16, position 11.2. And this is a cluster of genes on chromosome 16. And then each of these represents a different cluster of genes, like 15q11.13. There are these regions where, and I'll get into this later, explain it better, where the entire section can be either duplicated or triplicated or lost completely. So you have a variation in the number of copies of those genes. So it's interesting that autism can be caused by either just one gene being altered or whole clusters of genes. And I'll talk more about that later. Now, environmental causes of autism there are a few things, and I'm less familiar with this, to be honest. Genetics is my research field, but there are environmental, like toxin exposure or pathogen exposure. When the mother is pregnant, if they are exposed to certain things, it can affect the development of the child, and this can somehow be associated with autism. Again, not really well understood. Autism is a good field of science to be in because there are a lot of open questions, and obviously this is important work to be done. So exposure to pathogens, toxins, pollution, even stress, extreme stress on the mother, or infection. Another one that's interesting though is valproic acid. So this is a medication that's used for epilepsy and bipolar disorder. And if the mother is exposed or taking VPA during pregnancy, this can often result in autism. Then we have idiopathic autism. This essentially just means we don't know. The word basically means question mark. We don't know what is causing it. So there are certain people with autism that there's not an explanation of why. There's no genetic change. There's nothing. So, and that's again another big question. And lastly, vaccines do not cause autism. I'm just going to quickly explain this. So, message. Please take home, vaccines do not cause autism. What happened was in 1998, a scientist named Andrew Wakefield, I believe, was being funded. His research was being funded by a company that was in a lawsuit against a vaccine company. And so he fabricated data that showed that vaccines were associated with autism. It was totally fake, totally made up. He published it. And then later it was found out that it was fabricated. When the paper was published, the news, of course, hey, everyone, they publicized this, vaccines are associated with autism. And they were right to, because it was a scientific paper, it came out. But when it was retracted, when the paper was removed from the journal and no longer, it was discredited, the news articles or the news media sources didn't do a good job of clarifying that that wasn't breaking news. And so a lot of people were left continuing to believe that vaccines cause autism. Since then, many studies have been done that verify that, in fact, vaccines are not associated with autism. So, don't like to call people out or anything, but if there are any anti-vaxxers in here, I just, this is from a scientist that studies autism. This is the truth. I can promise you that. So here's a little time for a poll. Did you know that the link between vaccines and autism is unsupported? Is this news to you? I'm very curious to see what the results are on this poll. And do you want to take a quick intermission here?
If you guys just want to comment this down in the chat, any experiences that you have with this or what you just think about this in general, please feel free to comment them. and then we can just talk about this a little. I think for me, personally, I think my experience, I think so. I've always known that, just things like that. This is not news to me. That's great. We're really happy about this. We're really glad that you guys know that this is unsupported.
Great.
I think personally for me, I found that I knew it was unsupported, but when I was, came into contact with people who didn't believe the same thing, I didn't have that knowledge about why it was unsupported. So I think it's really great that you brought this to our attention.
Yeah, definitely. What happens to me a lot is I'll meet people and they'll ask me what I do, and I'll say, oh, I study autism, in neuroscience. And a lot of times people will say, oh, you should study why vaccines cause autism. And I'm just like, well, actually, let me explain that, that it's not something I'm mad about. It's just something I often address as an autism research scientist. Comes up a lot. So again, I can't see the chat. I don't know what's going on, but I'm gonna go ahead and move on to the next slide. Anulpa, you can please interrupt me.
Absolutely, yeah.
So next I'm going to talk about autism pathophysiology. And what this means is the disordered physiological processes associated with disease or injury. This is a definition from Google. Patho is typically associated with like disease. That's what that prefix means. Physiology is, what physiological processes in the body, are changed in that disease. So the first thing I'm going to talk about is altered brain cell proliferation and growth. So it's often seen that in children with autism, we have, in genetic models of autism, we have differences in the way brain cells grow and divide and branch out and communicate with one another. Some are, and sometimes we see that it's reduced, and sometimes we see that it's expanded. So here's a brain scan of a neurotypical child. Here's a brain scan of someone with microcephaly, and here's macrocephaly. So it's thought that potentially these changes in the way these cells divide and grow throughout development may be associated with autism, because we often see both microcephaly and macrocephaly in children with autism. The next thing I'm going to talk about is changes in gut microbiome. So this became hot news recently, last couple of years. I think it was like 2016. So it was discovered that there's this major link between your gut microbiome, which is essentially, the bacteria and stuff that you have in your stomach, in your digestive system, how this can relate to things like neurodevelopmental disorders like autism. So in this study, this one research paper that I'm taking this from, they took gut microbiome from children with autism and typically developing children and implanted them into mice, and found that the microbiota from children with autism induced social deficits in mice. So there's this big question of like, how is that possible? So this is the kind of an expanding topic of the gut-brain axis, it's called, and how the brain and the gut communicate. Because as a neuroscientist, I would think the brain has control over the gut, but the gut definitely can't have control over the brain. But that seems to be false. So just a general tip I'm going to share. As a scientist, I have always, you learn to be skeptical, but with experience and with being wrong many times for being too skeptical, it's a good idea to keep your mind kind of open to things, because science and biology have a tendency to blow your mind. So what's interesting about this is that they're now actually testing microbiota transfer therapy, transferring the microbiome from healthy, typically developing children into children with autism, and they actually are finding that it improves some of the symptoms of autism, which is, again, just incredible. It's amazing to me. Something I don't know very much about, though, again. And then another thing, it's kind of funny, people always bring this up. This is like one of the first things that people always mention to me is like, oh, there's this reduced regional connectivity, that different brain areas don't connect as effectively in autism. And so, I mean, it's a thing. This is definitely a thing. It's not like a huge, well-known, like this is the cause in autism, but for some reason people talk about it a lot. But this is a review article talking about it. And this is a scan where they're looking at some white matter tracts, and you could see that, here's a very strong regional connection between two brain areas, where in a child with autism, it's reduced. This is a mouse brain over here. And here they're just looking at general white matter tracts. I'm not even sure exactly what all this is, but you can see all the white matter. But by white matter, I mean axons, myelinated axons that are projecting between different brain areas to connect the two. And in the mouse model of autism, there's a huge reduction. So there is this open question of, is there less interbrain regional synchrony, or, are these brain areas communicating as well? Maybe this trouble with passing information from region to region. Now, lastly, the excitation inhibition imbalance theory of autism. I just wanted to show this right here because this is a well-known theory of autism. And I want to show this because when you Google this, or when you search this on Google Scholar, the database of research papers, you get 10,300 results for a phrase that if you read this, you would probably have no idea what this meant, or you'd think like, this is gibberish. So that, it's a very well-established thing, and I'm going to talk to you about it. What this essentially comes down to is in the brain, in the specific brain areas and within cells, you have excitation, which is cells activating one another and increasing their activity. And you have inhibition, cells turning each other off and shutting down the activity of one another through synaptic connections. This needs to be balanced. The two, excitation and inhibition, need to be balanced for the brain region to be functional. And we often see that the balance is shifted in one direction or the other. So let me just, I'll kind of come back to this and then I'll give a very detailed explanation. So the prefrontal cortex, and you may think this is coming out of left field, but the prefrontal cortex is really important for autism. And this is the brain region that I focus on. So everyone's got one. It's kind of right here behind your forehead. It's very important in higher level, what they call executive functions, things like decision making and complex thought and planning. It's very important for personality expression. People may have heard of Phineas Gage, the guy who a railroad spike went through his brain, but he survived, and it hit him in the prefrontal cortex, and he survived, but he was completely normal. He was completely healthy and everything, but his personality was completely different because he lost this brain area that is important for that. And it's also important for social behavior. As you might think, a huge part of social behavior is your personality expression. This is your way of communicating, your way of interacting with people. So I'm going to ask everyone to hold on tight and buckle in and try and focus on this part, because this is where things are going to get really a little bit more complicated, and I want to make sure that everyone gets this. So in the prefrontal cortex, there are two major cell types that I want everyone to understand. So first, there are pyramidal neurons. They're called pyramidal neurons because they're triangular. They look like pyramids. The cell body looks like a triangle. And these cells, here's just kind of a, whatever little schematic or a little graphic of them. These cells produce glutamate, a neurotransmitter. And glutamate, when it's released in synapses, and cells communicate with one another through little synapses where they release neurotransmitters onto receptors. Glutamate activates NMDA receptors and AMPA receptors. Don't worry so much about that. All I want you to know, and this will be clear on the next slide, is that these cells that look like this are producing glutamate, which activates the cells, like plus sign, and that'll be on the next slide. On the other hand, we also have interneurons, these kind of more branched out, they have like a central cell body, and then they have branches that release GABA, which is an inhibitory transmitter, and it activates GABA receptors, easy enough. So GABA, these interneurons integrate with these cells to shut them down and to shut one another down. So you can sort of maybe start to get an idea of this excitation inhibition balance. You maybe start to think, okay, maybe there need to be an equivalent number of these cells in this brain area for it to function properly. So here's where things are going to get a little crazy. I want you to step into this. We're going to zoom into the brain. We have some cells here. They're labeled. So we have cell A, B, and C. All right. And if anyone in here is a hardcore neuroscience person, I know this is going to be like anatomically incorrect when it comes to cellular, a lot of this stuff that's happening, but just let's simplify it and let's just look at how these cells interact in a brain circuit. So again, glutamate activates cells, GABA shuts cells off. So these cells produce glutamate. Let's say cell A and B both decide to release some glutamate onto cell C. This is excitatory. So this is going to cause cell C to activate this. I'm colorblind. I'm assuming this is green. Hopefully I got it right. This green background is supposed to indicate the cell's like kind of getting like hot and it's excited. It's activated. Now, so let's just say, don't be afraid. It's getting more complicated. We also have cells D, E, and F. Okay, and don't worry so much about them. Let's just think about cells D, E, and F as kind of another section. So we have cells A and B that are communicating with cell C, and then this message is getting passed down along to these next set of cells, D, E, and F. So since cell C is now activated, what happens in cells, and the whole purpose of synaptic transmission and these cells communicating, is a cell gets activated, it fires an action potential, and it releases its neurotransmitter. So cell C has been activated by A and B. Now it's going to release its neurotransmitter onto D, E, and F. This is in turn going to activate these cells, and the message is going to continue to get passed along. So you can kind of think about how all these cells, all in your brain, interconnected with one another. You can actually kind of piece together this idea of like all these circuits connecting and how they pass messages to one another, and maybe these project into different brain areas. It's all very interesting to think about. But so just to simplify things now, what's going on here is that cell A and B are talking to cell C, which is talking to cells D, E, and F. So we have a message being passed along. But we have our big bad interneuron here in the middle. And what this interneuron is going to do is it's going to connect with all of these cells. So I'm going to give you, this is an example of inhibition, and it's called recurrent inhibition, but don't worry about that. This is just kind of a real life example. This is something you might learn in a neuroscience class. So we have all these cells that are active. So now these cells are fired up. They're going to start releasing their neurotransmitter, glutamate, onto the interneuron. And this activates the interneuron. And if this is getting a little complicated, just try and think about like, at this point, every cell in this circuit is all activated. These are all firing all at once. And the reason that's important is because now that the interneuron's activated, it releases its neurotransmitter, which is GABA. And the result of GABA being released is that it's going to inhibit these other cells in turn and shut them off. So as a result of that, this cell C is now shut off. It's going to stop releasing its glutamate, which is going to allow these cells over here, D, E, and F, to slowly return back to baseline excitation levels. So what's actually happening here is that cells A and B are passing this message along, and cells D, E, and F are able to go back and shut themselves off by talking to this interneuron and saying, hey, tell those other cells up there, A, B, and C, to relax. Like, we got the message, okay? And so this is a kind of a feedback system. And so let me just pause here and ask, how's everybody feeling about this? This is on the next slide, but I want to know, how's everybody feeling? Do you understand, does this make sense to you, how a circuit would be built this way and designed this way?
So if this makes sense to you, you can just say yes in the chat, or if you have any questions or if this doesn't make sense to you, you can say no, and then we can kind of go over this again a little. So again, yes if it makes sense to you, no if it doesn't, or write down any questions. All right, I'm seeing some yeses, that's good.
Okay.
All right, so I think most people are keeping up with this, all right.
All right, excellent. So now, you may be so sick and tired of this, but I'm going to keep going because it's important. I want you to now think about an individual cell. We're going to zoom into a cell. So here's the cell body. And by the way, I'm explaining this way because it's not really relevant it's not extremely relevant to the talk. It is. You don't need to know this, but I'm choosing to explain this because I think it's a cool lesson to learn. I like this component of neuroscience, is thinking about the excitability of cells. So here's a cell. It's expressing NMDA receptors. Don't worry about it, the details, but they're activated by glutamate. Sorry. So they're excitatory. This is going to cause, they're going to open their ion channels. You have calcium that flows into the cell. Let's say you also have AMPA receptors. These are also going to be activated by glutamate. So this is going to cause sodium to flow into the cell. This is actually, sorry, ignore that too. This should be Na+. Not that anyone cares. So now you have these excitatory receptors being activated. So if we're going to look over here, and I'm assuming everyone can see my cursor because I've been using it this whole time, and hopefully you can see it.
We can, yeah.
Okay, good. This is going to cause the cell's membrane potential to increase. So this is not good. The cell does not want to be, it must activate at some point. But if this were all that was happening and the cell was just active, this is unhealthy for the cell. And in fact, if you think about this from a more broad perspective, if all the cells in the brain were like this could be associated, or this could be causing epilepsy, because when the cells can't shut themselves down, that's how seizures occur, is this spontaneous, uncontrolled activity in the brain. So the reason we have GABA receptors, which is, remember again, interneurons release GABA, is so that GABA can come along, activate them. They allow chloride to flow into the cell. Don't worry about it. And this causes the cell to return back to its normal level, and the cell is pleased. It's happy. It's relaxed, and everything's back to normal. Now, on the other hand, let's say there's a lot of GABA in the synapse, and there's not as many of these receptors over here. these AMPA and NMDA receptors to keep the cell active. Now the cell is going to slide down to too little activity. All right, what I was in the middle of saying, I was explaining this really complex, again, one of my super complex graphics, but the end result is what I was saying is that it's important that the balance be maintained, the activity balance be maintained on a cellular level, because in the end of the day, this brain region, the prefrontal cortex, is involved in so many different functions. So top-down guidance of attention and thought, inhibiting inappropriate actions, regulating emotions. And it's, I think about the prefrontal cortex as the mother of the brain and all the other brain areas as the children of the brain. So the mother of the brain is kind of like, oh, you know, like making sure everyone's in place, everyone's doing what they're supposed to, just kind of regulating everything. And you can imagine how having too much activity or too little of activity in this brain area can cause a lot of different changes, because it's not just restricted to this one region. It's affecting the entire brain, how all these different functions are operating. So so now I was going to ask a poll. Did you learn something? Sorry, I'm like replaying, like what happened? Because I was talking to myself. Did you learn something, or are you confused? I'm mostly wondering if you learned something. I would love to know.
Again, so same as the crowdcast, we can all just comment them down in the chat.
Let's see if I can get the chat to show up.
So if I can see yes, no, specifics about what you learned, what you're confused about.
Or if you have any questions.
Okay, I'm seeing some yeses. Yep.
Oh, here we go. All right, cool. All right, I'm going to move on. Interrupt me if there's any questions that come up. So now let's talk about animal models of autism, specifically mouse models. So there are many models of autism. And what I mean by that are there are transgenic, and what that means is genetically modified, essentially, knockout mouse models, where, remember in the beginning I explained there are hundreds of genes that their mutations are associated with autism. Those genes are identified through genetic screenings of huge populations of people with or without autism to find, you know, what gene mutations do we see often. We can then deliberately have a mutation of that gene in a mouse and test whether or not it affects their social behavior. So this is a huge part. I mean, this is what I study. So if you have questions about this, please let me know. But these are just a few of them, like SHANK3, CNTNAP2, FMR1. These are just very common genes that are mutated in children with autism that have been used to develop very viable mouse models of autism. And you may be thinking, mouse models of autism, what? But I'll get to that. There are also copy number variations. So there are mice that carry these same duplications or deletions of those gene areas that I was talking about, or those chromosomal regions. And actually, amazingly, a lot of those are conserved. Like mice have the same exact region with all the same genes in the same order that we do, which is very lucky. It makes it great for studying these genetic changes. There are environmental models of autism. So you can actually, like if you expose a mouse in utero to an immune response in the mother, this can actually cause the pups to develop autism. I'm sorry, there's no such thing as autism in a mouse. This is also a very kind of new thing in science, is that we can have the mice develop autism-like behaviors. So remember also I talked about valproic acid, VPA, that if a mother is exposed to it, this is also true in mice, which is, again, super interesting.
Stanley's growing trees.
Okay, so now let's talk about 16p11.2 copy number variation. So this is what I said in the beginning. I said, okay, I'll explain it a little bit more. So 16p11.2, again, is this section of chromosome 16, position 11.2. This is a very sophisticated drawing of it. And there's about 27 genes wrapped into this area. And like I said, it can be either deleted or duplicated. So literally the region is either lost or the region is duplicated and you have two copies of it. And of course, there's also another copy on this chromosome because we have sister chromatin, they're identical. But you can either have just a duplication or a loss, and either the deletion or the duplication are both associated with autism. But there's this huge question of why. So I wanted to bring up this example. So Down syndrome, trisomy 21, is very similar to a copy number variation. What Down syndrome is you have a third chromosome 21. So this is kind of the same thing where I want you to think about the duplication as that, where you have this extra section of chromosome, but this is associated with autism. Now, the 16p11.2 duplication can be, again, replicated or reproduced in a mouse, and people have done that. I didn't do that. Another lab created these mice, and of course, a big question is the benefits of mouse models. It's important to be able to really screen what's happening on a biological level and look into the brain. It's, this is not available for us to do with human brains. So it opens a lot of doors, and it's super important. But a question I often get is, how can you tell if the mouse has autism? And again, that's not the proper phrasing, but how can you tell if a mouse has any changes in their social behavior? So I love talking about this. I love animal behavior. I get so intrigued by it. Like watching animals outside, I'm just like, whoa, like what is that bird thinking? And so I love to design these paradigms for testing specific behaviors in animals. I just kind of like have this connection with them. I don't know. So one is the 3-chamber social preference test, where I don't want you to look at this and think, oh my gosh, this poor mouse is behind bars. It's just like this little cage. It's very comfortable. But we have this apparatus where it's just a big open square rectangle. And on one side, we have a mouse under a cup. And on the other side, we have an object, a wooden block under a cup. And the reason they're under a cup is not because they're in jail. It's because they need to be restrained so that the test mouse can choose whether or not they want to interact with either one. So in science, people that study neuroscience or whatever in mice, there's something called wild type. Wild type. This is your healthy, totally genetically unaltered, like inbred mouse, where everything is exactly the same, no matter what. Every lab in the world has the same exact mouse. There's no differences. So this is your control mouse. So if you test a control mouse in this test, they are going to run around just like this and spend a lot of time interacting with the mouse, whereas a mouse with one of these gene mutations may prefer the wooden block. So again, I mentioned the 16p11.2 duplication mouse model, and I know that's a mouthful. I'm going to call it 16p duplication or something like that from now on. If you look at the, again, wild type, WT, so this is actual data. This is my data. This is a heat map from above. So we're looking at this, and on the left, we have the mouse, the social stimulus. On the right, we have the object, the non-social stimulus. And this is over a 10-minute period. So you can see the wild type mouse spent a lot of time. The red is more time spent there. They spent a lot of time interacting with the social stimulus, not so much with the non-social. So they clearly preferred interacting with the social stimulus. On the other hand, these mice with the 16p duplication, you can see they spend a little bit less time. They're not so engaged with the social cue, and they also spend more time with the non-social. So it's kind of even. They almost kind of even out. They don't really show that strong social preference. On the other hand, we have the social approach task, where this is just, we put a mouse in the middle and we measure how much time they spend interacting. A wild type will do lots of interacting, whereas the 16p or an autism mouse model may spend more time kind of in the corner or a little bit more reserved. So again, here's our actual data. Wild type mice spend plenty of time interacting. They're, circling, sniffing, really engaged. The 16p mice kind of stick to the corners. They don't really like to go in the middle and interact so much. So, and again, this is my data. So from here on, we're going to be talking about my research in the context of everything I've been talking about. But just to clarify, in case anyone is uncertain, so I'm a PhD student, and I've been studying the 16p duplication and kind of characterizing it and figuring things out how this works. And I'm just going to kind of lead you along that story on a scientific level. So now we're moving away from the clinical stuff and doing more of a scientific presentation. And it's going to get scientific real quick. So, okay. Again, remember I mentioned this prefrontal cortex brain area is super important for, the activity levels. We have to be balanced, and it's important for social behavior. So since we saw this we call it social deficits in mice, we wanted to investigate how the prefrontal cortex was functioning. So this is just, if you were looking, this is like the front of the brain. Like if this is my brain, we're looking at one half, and this is a mouse brain. Like obviously this is a drawing, but we have the anterior cingulate. These are just the prefrontal cortex, and we measure the activity of these cells through something called slice electrophysiology, which don't worry about it. The details, the technical details of it. But this is a little bit complicated, but what we're doing here is we're stimulating the cells at varying and increasing intensities, and we're recording the output of what the cell is doing at these different intensities And this is NMDA receptors and AMPA receptors. So if you just look at, these are actual traces of the current of the cell changing. This is like current ionic flow, again, whatever. And you can see that the amplitudes of these current changes are completely identical. So there's no change in the NMDA, the glutamatergic stuff, remember the plus sign, there's no change in this. But when we activate the cells and cause GABA to be released, we find that the amplitude on the cells that are receiving the GABA is reduced in these 16p duplication mice. So you can see there's a pretty pronounced reduction in the activity. And again, so GABA is inhibitory. GABA shuts off the cells. But so we're seeing a reduction in the inhibition, if that makes sense. So instead of, they're not being shut off as effectively as they should be. So they're staying a little bit more active. And what we see, which later turns out to be correlated with the GABA changes, is that when we actually just take a cell and we activate it a little bit and it starts to fire action potentials, we don't see, or we see an increase in the number of action potentials that the cell is firing. So these cells are more prone to activity, essentially, is what we're seeing, is that the cell's excitation and activity level is increased. So again, we're thinking about that, what I showed you before, that activity that needs to be balanced. These cells are clearly on the high end of this. And interestingly, 16p11.2 duplications, human patients with this genetic duplication often have epilepsy, which makes sense because we find that the cells are too active. So now, remember our good old interneuron? I wanted to see if the reason we're seeing less GABA activity is because these cells just aren't there. So I did some immunostaining, and so here we go. We have this rectangle here. We're looking in the prelimbic cortex. So this is actually images, and basically these little dots, these circles, these are the actual cells. So this is what these guys actually look like in a brain. I just wanted to show. It's kind of neat. But we counted the number of cells, and they're about the same. In fact, maybe a little bit of an increase. So it's not because there's a loss of these cells. That was our first question. So we were a little bit lost, and we thought, how can we figure out what is causing, we have 27 genes that are duplicated in this mouse, and we have this one change in synaptic activity.
Just to interrupt you, I think we have a question in the chat. So someone asked, what are PV interneurons?
All right. Okay. Yes. PV interneurons. So remember I was talking about, we have the pyramidal neurons and we have interneurons. There are different types of interneurons. And one of them is, sorry, by the way, I copied this from my interviews as my postdoc presentation. PV is short for paravalbumin. It is a gene that only these types of cells express. So we actually stained for this protein, paravalbumin, to identify these cells. So it's just a subtype of interneuron that's very common in the prefrontal cortex. So hopefully that addresses that. Please interrupt me if there are more questions. So we had to figure out how this duplication of these 27 genes is associated with this change in this one type of synaptic activity, which is a huge question, especially for a first-timer PhD student trying to figure out how to do science for the first time. So to get a little bit of help, we did something called RNA sequencing, which is basically what we do is we take the prefrontal cortex and we sequence every single gene and look at which genes are being expressed at higher levels and which genes are being expressed at lower levels in this brain area. And so this is a volcano plot. All the red dots, so from right to left, this is increased expression, decreased expression. You can see the red dots indicate genes. Every dot is a gene. The red ones are all genes that are significantly different in these 16p mice. So you can see there are a good amount of upregulated increased expression genes and a whole bunch of decreased expression genes. So I looked at, I actually got a list of genes that are changed, and I looked at it. And as I was scrolling through, there's hundreds of them. This is a good section of my PhD. I found this gene NPAS4, and I looked it up, and it turns out that it's called neuronal pass domain containing 4. Don't worry about that. But it's an activity dependent transcription factor. What that means is it's activated by activity. When the cell's active, NPAS4 is active, and it's a transcription factor, meaning it goes into the nucleus and it controls the expression of other genes. And more importantly, it regulates the formation of GABA synapses. So here we have a gene that builds these GABA synapses, and it's reduced. There's reduced expression of it, and we have a loss of GABA, signaling. So this is probably, maybe this reduction in this MPAS4 gene is what's actually the cause here. Anyone have any questions? Good. Okay.
I don't see any questions now, but if we get any questions, please comment them in the chat. Love the chat. That's all good. And then we can talk about it. Yeah, for now we're good.
And I know that this is probably getting a little bit less interesting for you because we went from like really crazy autism stuff to like all of a sudden we're looking at a list of genes, and I'm very sorry about that, but it'll get more interesting in a second. So I of course wanted to verify it's important that MPAS4 was actually downregulated. What I mean by that is the expression level is decreased. And it is, in fact. So this is, I looked at the level of the mRNA, which is messenger RNA. It's the expression of the gene. And we found that it was decreased in the prefrontal cortex of these mice. But what's very interesting is that we also found that it was decreased in human prefrontal cortex of children with autism. So we actually have human brain tissue in the lab, and we use it once in a while to test these types of things. And it is decreased, which is interesting, because this may mean that, and these are also not, the brain tissue is not from only those with the 16p duplication. This is all different types of autism. So this may mean that MPAS4 is important beyond just this one gene mutation and more broadly in autism. And this is something I plan to investigate more in my career. But I'm sure everyone's happy to see this again. Let's talk about what this really means. To be more simple. So the cell's activated. Don't worry about the details, but the excitation goes up, right? We see over here on the right, the cell's activity goes up. Now, cell's unhappy. This causes, if we look here in the nucleus, this is going to cause MPAS4 to be expressed. It's activity dependent. MPAS4 then goes and builds GABA synapses. GABA synapses are inhibitory, and this allows the cell to return to its normal level and be happy again. So it makes sense. This is it's a feedback loop. So in the 16p duplication, what I theorized was happening was that the cell's activity was normal. Remember, and we showed that NMDA and AMPA receptor activity was totally normal. So we know that's fine. Cell gets excited, it's too active, but normally MPAS4 would be expressed here, but it's not for whatever reason. And as a result, we don't have GABA synapses being formed. As a result of this, there's fewer GABA synapses. This causes the reduction in the GABA activity that we see. And this potentially also causes, up here we have the cell that's way too active. This is why the cell's also firing too much. So it makes sense. It's a perfect explanation. So I thought, just maybe, if we can cause these brain cells to start expressing MPAS4, will this have a dramatic influence on their behavior? So again, just ignoring the major details of this, we developed a virus. Well, actually, we purchased this virus. And I'm sure when you hear virus, you're like, what? Like coronavirus, but it's not. It's basically, this is used often in neuroscience, that people use viruses to deliver specific messages to cells, because viruses are actually really great tools for neuroscience. So this particular virus, it's not dangerous or anything. It's not going to make you sick, but it is designed so that it carries this message to overexpress MPAS4. So it's causing the cells to boost up MPAS4. So I was able to inject this into the prefrontal cortex of these mice. And this is like, I know this probably sounds crazy, but this is actually like a pretty mild brain surgery for a mouse, and they are perfectly fine. Like it's really not bad. This type of surgery happens all the time for people. Like if they have a brain bleed and they have to like drain it or something like that, it's pretty simple. So the mice are perfectly fine, but we can then look at what's actually changing in their brains. So this showed that MPAS4 was increased. It did work. The virus did what it was supposed to do. And now what we're looking at here, and we have 4 bars. So we have, this is our wild type, our control group. Then this is our 16p duplication. What we're staining for is VGAT. VGAT is a vesicle that transports GABA at synapses. So what we're actually seeing here is, this is a marker for presynaptic GABA synapses. So every one of these little speckles, so let's look at the wild type plus GFP. I'll explain that in a second. Every one of these little dots represents one GABA synapse, and every one of these bigger blue circles is the nucleus of a cell. So you could tell these things are everywhere. Like these synapses are all over the place, because of course they're super important. And now you can kind of imagine that graphic I showed you from the beginning where cells A, B, C, and D and all that are interacting, this is happening everywhere. So that's kind of a simplified version of like this crazy circuitry that's going on in the brain all the time. I saw that there was a message, but I can't access it now. Oh, I can. Okay, so GFP is a control virus. It's green fluorescent protein. So you see how these cells are all highlighted in green? This means we just injected the mice with a control virus where it changes nothing. So some mice got a GFP virus just to control for the actual surgery and make sure it didn't have any effect. And other mice got this NPAS4 overexpression virus. So if you look at the GFP groups, these are our control groups, you can see that in the mice with the 16p duplication, there is such a dramatic loss of these GABA synapses. They're sparse. They're almost not even there, which makes sense and is kind of along with my theory that NPAS4 is not doing what it's supposed to and causing these synapses to be built. But when we inject this NPAS4 virus, where's my mouse? You can, comparing these two, you can see there is an increase in the number of synapses. It's not as dense as what we see in the control mice, but there is an increase. So of course, the big question is, we've added back these GABA synapses. Is this going to have a functional impact on the way these cells actually activate and communicate? So again, looking at our electrophysiology, this is kind of complicated. Sorry about this figure, but let's just look at this light blue. This is our baseline of the 16p duplication mice. And then this is actually navy, but it looks black. This is after the injection. And you can see that the level of these, the amplitude of these GABA responses is much bigger. So here's our control. This is a pretty large response. In the 16p mice, there's a reduced response. But after the injection, you can see that the response is enhanced. So that's good. The same is also true for the action potential firing. So it reduced the hyperexcitability of these cells. Don't look too much into this figure. It's complicated, but it seems like this is fixing the change that we saw before in the actual cellular activity when we record it. Any questions? I'll give it like 1 second. Okay.
I don't think we have any questions now, but please, if you do have questions, keep them coming. Oh, sorry. So someone asked, what is the summary of all of that?
Okay, great question. The summary of all that is that and this is about to get a lot more interesting in 1 second. The summary of all that is that by restoring this expression of this one gene in this mouse with these 27 genes all duplicated, by just changing the expression level of this one gene, we are able to restore the GABA synaptic transmission. And we're able to allow those interneurons to do their job and restore that balance from being way too high to now being back in the middle where it's supposed to be.
And we have another question. So they said, so 16p plus NPAS4 equals better responses in neurotypical mice and humans? Is that the general summary of your research?
So 16p is the genetic variant. So it's not neurotypical. 16p is the autism model. Giving the 16p mice NPAS4 brings them much closer to the neurotypical. I guess that's hopefully addressing that question. It's definitely not like better. It's not better than neurotypical. It's just that we are basically seeing is in this genetic mutation that we know is associated with autism, we're seeing a change in the activity of these cells that makes sense considering this theory of autism that it could be too active or too inactive. So we're seeing that, okay, they are in fact too active. And by targeting NPAS4, we're able to restore the activity to what we would expect would be a more healthy level of activity that's not going to be. Okay, I'm just reading this. Yes, in fact, sorry, mistyped neurotypical. So 16p plus NPAS4 is better cellular responses in the ASD mice. Yes, and now this is going to be the most important part. I'm going to skip ahead. Here we have the social test again. So this is our control wild type mice. This is our 16p duplication. This is a social preference index. So this is basically a measurement of what % of their time did they spend hanging out with the mouse over the object. So you can see the wild type mice are above 50%. They highly prefer the mouse, whereas the 16p duplication mice are kind of like even split, like I said in the beginning. But after injecting NPAS4, these mice become almost as social as the wild type mice. And you can see, so here is your 16p plus GF, but this is your baseline 16p duplication mice, about even split. But after the NPAS4, they're spending a bunch of time over here with the social cue, not so much with the non-social cue. So this is the real important part of this, is that by restoring this homeostatic, I would say, or this kind of balanced activity of these cells, this is sufficient to cause these mice that have social deficits under normal conditions to now become much more social, which would indicate that those cellular changes are a critical component of the social changes that we see in these mice, and may be critical or important as well to the human patients that have the same genetic alteration. So, little poll, do you think, based on this, that targeting MPAS4 could be a feasible treatment strategy for autism? And I'm glad I put this poll in, because it's a reason for me to explain that I injected a virus into the brains of these mice. That is not a feasible treatment strategy for anybody with any condition. But what my study accomplished is it identified a molecule that may be worth considering targeting through like a normal drug, like, you take a ibuprofen, you have a headache, that type of thing, or you take a SSRI, like people who have depression. It's similar to that. Can we find some sort of compound like an SSRI that targets NPAS4? So this is a really mechanistic study, in that it's not like translational. We're not treating children with this virus, but it identifies NPAS4 as maybe a target. So I want to know if you think it could be targetable.
So we have two questions in the chat. The first one is, can we duplicate this surgery into ASD human patients as well?
No.
Okay.
Yeah, I mean, theoretically, yes. Theoretically, we could do the exact same thing. But the problem is, like, it's just, it's brain surgery. Nobody is going to want to say, you know what, yeah, okay, my 10-year-old son here is just, like there's no way. But we may be able to, like I said, find some sort of drug or compound that that can simulate the change that we have with this virus injection. I think that question, feasible treatment strategy. Yeah. Right, of course. It is a question. So the thing someone said, I think that question about the feasible treatment strategy raises an issue of ethics. Exactly. I mean, there's a huge ethical question of, can we do an injection, a brain injection to someone? Of course not. So, and also this is an important thing for me to say, is that there are no treatment strategies, pharmacological treatment strategies. There are no like drugs that you can take like an SSRI for the social symptoms of autism. There's nothing. We don't know anything about how this may be treatable. And many people with autism like having autism. I've learned this over time through my work, is that many people see it as like a superpower, and it's amazing actually. But there are also a lot of people who have autism along with epilepsy and intellectual disability and all these, like I said, it's very comorbid. And for those people, it can be very challenging. It can make their life very, very challenging. And so what we want to do is, we don't want to cure autism. That's, a lot of people say like, oh, like how could you? But we just want to try to identify some way so we can create some sort of treatment to be available for people who do want it. So great question. What can stimulate NPAS4 in humans? That is what I'm currently working on. So NPAS4 is activity dependent, as I said. So it would make sense that, okay, well, what if we activate these cells to try and activate NPAS4? But the problem is the cells are already too active, and people with this mutation have seizures. So we don't want to activate their cells and start causing seizures in these people. So we need to figure out, and I didn't include this in this talk because it was too much, but we need to figure out the cells being activated, but for some reason NPAS4 is not being expressed the way it should be. We need to figure out what is upstream, what molecules are controlling NPAS4's expression, and see if we can target that. And that's exactly what I'm doing right now. And I did find that upstream of NPAS4 is a molecule called HDAC5, and I'm not going to get into it, but I'm now testing whether or not I found a compound that inhibits HDAC5, and I'm trying to see if I can do the same thing, but without like taking the virus injection out of it, and instead giving the mice like a supplement. So And that would be translationally feasible. So let me finish up here. One quick thing. I almost took the slide out, but I'm just going to run through it really quick. The 16p, remember I said there's a duplication and a deletion. So this is another paper of mine from 2018 that we found that in the deletion mice, the opposite direction, they have a reduced activity. So here's the cell firing, here's a control cell, here's the 16p deletion cell. But we found that if we were able to activate these cells, so I'm not going to get into the details of how we did that, but if we can restore the activity of these cells, it made them significantly more social. So it looks like we have the duplication on one end causing increased activity, the deletion on the other end causing decreased activity. And in either condition, if we can sort of neutralize that back to the middle, that is sufficient to improve the social deficits that we see. So on that note, I'd like to acknowledge all my acknowledgees. Of course, I have to thank funding and NIMH and Nancy Larry Marks Family Foundation. Not funding to me, but to my PI, Dr. Zhen Yan, who is right here. A lot of my lab members, awesome for teaching me the information that I shared, for teaching me how to do the experiments, and for helping me with these experiments. And Michael Greenberg at Harvard for helping me provide a reagent. And with that, I will open it to Q&A.
All right, so I think we've answered most of the questions that people have had in the chat so far, but if you have any more questions, please just comment them down below and we will get to them. But right now, I want to get to some of the questions that we had entered in our Crowdcast page. So someone asked, I heard that this is about the diagnosis of autism. So they said, I heard that it is sometimes harder to diagnose females with autism due to masking behaviors. Could this be skewing the data to appear as though males are more likely to have autism?
So again, I'm not a doctor, but yeah, I mean, I think it's possible. I think it is. I think it's definitely possible, but the question to me is it enough that it could be a 4-to-1 difference? I also, so when like I said, I sat in a diagnostic autism center, and I watched an assessment, and it was a girl. She was a female, and personally, and this is super anecdotal off of one experience, but I could very much tell, like, okay, there was no masking for me. I could see that it would be expressed in similar ways as if she were a boy. But so yeah, I think that is definitely a factor. But again, not a doctor, don't know enough about it to really give a true, thorough answer. And you should not value my answer because I don't actually know.
All right, so thank you. And another person asked, what was the hardest part of your research?
Oh, I think the hardest part of research is, and I'm just going to take this question as not just the actual research I did, but just becoming a scientist in general. I think the hardest part for me was this fear of learning these techniques. For example, performing a brain surgery on a mouse. And it's not a brain surgery I had to do once or twice. It's a brain surgery that I have had to do probably over 100 times. And it's like, you need to learn these very intimidating skills, and it's not as bad as you think. So I think my hardest part has been struggling with that personally, is my fear of being inadequate to learn these things and perform them. So, but the research itself was also, I mean, there were many times where experiments didn't work out and I just got lost and didn't know what to do. And that's why it's important to have good people around you in the lab and a good boss, a good PI in the lab to help guide you.
And this is our last question. I think this is really important to address. They said, so is there a chance that autism can be cured? And I think the word cure is really essential in this. So if you could talk about that.
Yeah, and by the way, so it says remaining meeting time 3 minutes and 9 seconds. So I'm going to get through this. Cure is a word that doesn't exist in science. Typically, treatment is a word that is used, and I do think that I believe that within the next 5 to 10 years, the first treatment for the social symptoms of autism will be available. Probably 10 years, and but the thing is, and I can tell you, you might be, I don't know how everyone's background is, but you might be seeing this and thinking, wow, this is incredible. This guy was able to make these mice with these social deficits behave very socially. But the truth is, this has been done so many different times in so many different mouse models of autism, and we still don't have a treatment in humans. So the translational capacity of these types of studies is limited. And a lot of people say like, well, yeah, of course it's mice. The truth is actually a lot of other treatments that work in humans have come from mice. Autism just seems to be a really complex disorder that, this is one genetic mutation that's associated with it. Even if I can find a treatment that works every single time, it doesn't mean it's going to help people with other mutations. So it's just a huge challenge, and autism is a tricky field.
Yeah, and just like, like you were saying, I think it's really hard to find, even treatments. But again, we just want to acknowledge that autism is not necessarily something that has to be cured. So I think
Yes.
that's just another thing to address. But we're coming to the end of the meeting. So thank you guys so much for joining us today. We hope you learned something and just enjoyed this presentation. Also, thank you so much for bearing with us during all the technical difficulties. So make sure to check out Ben's Instagram, which is linked on the screen right now, and also make sure to check out our Instagram at SciMed in Action. We've got a lot more cool events in store for you this year, so keep an eye out. And once again, thank you so much, and thank you for staying engaged during the technical difficulties.
Yeah, and I skipped to this slide because I thought my email was on it, but it's not. So I put my email in the chat if anyone has questions about it. So please feel free to reach out, and I'll look forward to receiving any questions you may have.
All right, thank you guys so much. We hope you have a great rest of your day.
Happy Sunday.